Chapter 12, Phlebotomized Sandflies, the Psychodidae. For the introduction. So the book discusses the challenges in studying the biology of phlebotomized sandflies, which are vectors of leishmaniasis and other diseases in tropical and subtropical regions. So the difficulties include small size and inconspicuous behavior, high species diversity but morphological similarity, priority of larval stages in the field, and difficulty in rearing. So phlebotomites are also known as sandflies. They're often confused with a different family of insects. However, the phlebotomites are found in forests from the southern United States to northern Argentina and are typically 2.5 to 3.5 millimeters in size, with a silvery gray to black color and covered in narrow, erect scales. They have a distinctive V-shaped wing position when at rest, and they move by hopping. So little is known about their eggs and the larvae, but laboratory studies suggest they prefer organically rich, dry soils. Female phlebotomites require blood for reproduction, and their bites can be painful, leaving a small pool of blood and potentially causing red swelling. While they are not strongly attracted to humans, they can adapt to paradomestic or urban environments, making humans an auxiliary host. The lack of species specificity in hosts has hindered the development of vector parasite model systems, as a single species of Cutaneous leishmaniasis may have multiple suspected vectors associated with it, making epidemiological models relevant to other vector parasite pairs. But these challenges have hindered research on phlebotomy biology, resulting in limited understanding of the behavior, host preferences, and larva habitats. So the text suggests 4 areas of research to improve our understanding of phlebotomy biology and its relationship to leishmaniasis epidemiology. So, which is 1, species distribution studies, molecular systematics, host preference determination, and larva habitat characteristics. For the general identification, the Psychodidae family, which includes phlebotomites, is ancient and has distinctive characteristics such as a narrow scales on the head, thorax, legs, and wing veins. So the phlebotomites are further distinguished by their elongated and fragile structure and piercing mouth parts, which are capable of taking blood. The subfamily Phlebotomites is divided into Old World and New World, as shown in the model. The taxonomic keys and distribution maps have been compiled for the Old World genre, while the New World genus Lusmia has been extensively studied and described. So species differentiation is primarily based on adult morphology, including male genitalia with the female spermatogonia and antennal and leg morphology. So around 90 characters have been identified as effective species descriptors, but long-term preservation of specimens is crucial for taxonomic study, and mounting individual specimens on microscope slides is essential for identification. The taxonomic nomenclature of phlebotomites is undergoing significant change, with efforts to revise the nomenclature based on morphological and molecular characters. Cladistic analysis and molecular phylogeny have supported the reorganizations of genera and subgenera, and further development of molecular methods will provide objective support for the taxonomic reorganization. Now for the next slide. This is the same images as in the previous slide. Just feel free to pause the video and observe them. For the next slide, the life cycle. So the life cycle of phlebotomite sandflies takes 6 weeks or more to complete. Adult females lay single eggs on crevicid substrates, which hatch in about 4 to 10 days. The larvae go through 4 inserts over 4 to 8 weeks, followed by a 4 to 6 day pupal stage. So the adults emerge, mate, feed on blood, and lay eggs, with females surviving long enough to potentially infect a second host. The laboratory observations have described the immature stages, while attempts to locate adult emergence points in the wild have had moderate success for only one domestic species, the Longipalpia. Now for the eggs specifically, so retrieving eggs from the field is nearly impossible, so information about them comes entirely just from laboratory observations. Females typically lay 20 to 50 eggs on rough humid substrates. Laboratory studies have shown that eggs can be retained and oviposited without fertilizations with high frequency. The plaster of Paris, when solidified but damp, provides an acceptable texture for many species to successfully colonize. The egg chorion has a distinctive structure of ridges, cells, microvilli that show species-specific patterns as observed through like electron microscope patterns. Now for the second stage, the larva of phlebotomide sandflies progresses through 4 instars, growing from 2 millimeters to 10 millimeters in length. The larva cuticle is thick and scleroticized to prevent water loss in arid environments, and the body surface is venose and darkly colored. The respiration occurs through abdominal and thoracic spiracles, and in later instars, and nutrition for larval growth is likely highly organic soil with substantial fecal deposition and decomposition. The complete life cycle is known for only about like 5% of species, and larva development can take from a month to 3 months depending on the temperature. Seasonal cycles in fly density suggest that larvae can estivate or enter this pause during in-hospital seasons. Now for the third stage, the pupa. Before pupating, the larva becomes sluggish and tends to move to higher, drier areas. The fourth instar, exuvium, is attached to the posterior segments of the pupa, which initially appear pale and cream-colored but darkens as it hardens. The pupa is relatively immobile, only like wiggling slightly in response to like probes, and remains in the state for like 4 to 6 days before the adult emerges. Now for the adults, adults move by hopping with wings held in a V shaped over the abdomen. Males and females are similar in size, but can be distinguished by their body shape and weight. Females have a round, robust abdomen and weigh like approximately like 0.6 milligrams, while males have a slender abdomen and weigh approximately 0.4 milligrams with a distinctive C-shaped terminali in side view. Now for the next slide, habitat diversity. Phlebotomy and sandflies can be grouped into paradomestic, associated with human activity, and feral, independent of human activity. Habitats. Paradomestic adults flies are found in high densities near larva habitats such as soil and feces mix, and are often collected from domestic animals, whilst feral environments are less predictable but can be found in moist forested areas, protected cavities, and caves. For the dispersal, adult phlebotomites are weak filers, dispersing less than 60 meters, with only 3% flying beyond 300 meters. This limits their ability to colonize new areas or recolonize areas that have been abandoned or treated with insecticides. Field capture of adults allows interference of or inference of nearby larva habitats with density peaks of nulliparous females indicating larva areas. However, predicting the presence of a given species can be difficult if their preferred environment is patchy or transient. Forest fragmentation leads to disjunct fly populations, making recolonization of forest patches more difficult in the event of like local extinction. Now for the sampling, phlebotomy diversity requires a variety of techniques for retrieving these specimens from the field. Some sampling methods for phlebotomy and sandflies include carbon dioxide-based CDC light traps, which are widely used, productive, but easy to use. Hand aspirator, they're labor-intensive, time-consuming, but allows for direct association between larva habitat and resting sites. The Shannon trap, which are effective in forests, attracts flies to light and collects them with hand aspirator. Now, each method has advantages and disadvantages. So the light traps, including the Shannon traps, they have the advantage of standardized sampling, but may not collect all species if some are not attracted to light. Now, the hand aspirator is mobile, allows for targeting searching, but may skew estimates of species composition if used alone. Now, using multiple methods consistently throughout the year is essential to approximate species diversity and densities in a particular area. Now, as for sandfly genetics, studying sandfly genetics is difficult due to their short life cycle, hard colonization, and diverse species. Even with established colonies, genetic models are limited, and no species has formal genetics or linkage maps. A dominant phenotype, two abdominal pale spots in males have been used in crosses, but hasn't been linked to other markers in lab crosses. Research focuses on cytogenetics and population genetics with implications for taxonomy. So cytogenetics attempts to extract folate chromosomes from sandflies have been unsuccessful. However, karyotypes have been established for 9 Lustemia and 4 phlebotomous species using mitosis and larva brain squashes. The chromosome number ranges from 3 to 5 pairs in phlebotomous and 3 to 4 pairs in Lustemia. Heteromorphism in sex chromosomes, chromosome number, and arm length can provide clues for subgeneric species relationship. Intraspecific heteromorphism has been observed in geographic stains of Longihalpius. However, meiotic events have not been described due to the small size of the chromosomes and lack visibility at the light microscope level. But Longihalpius, meiosis, and spermatogenesis begin with hours of adult emergence. Population genetics. So sandfly species have been studied extensively for their population genetic structure. Phlebotomys papatithi shows typical genetic variability across its range without strong evidence of local differentiation. Longihalpius exhibits varying degrees of allopatric differentiation with multiple studies indicating diagnostic characters separating different populations. However, the comparisons do not always cooperate each other well due to their opportunistic sampling. Field studies with careful sampling design and multiple analytic methods are needed to reassort the current evidence, though. The Whitmani does not show similar genetic compartmentalisms, despite a similar broad distribution, and the Shannon is found in tropical and subtropical forests, and they have its broad geographic distribution, but its U.S. population is isolated from its conjoiners by several hundred kilometers, with a high degree of differentiation in allosom constitution, but without morphological divergence. The Andes Mountains appears to have provided a barrier to gene flow, separating populations in the northern and eastern river basins from those of the Orinoco and Amazon basins, according to the book. And now for the human diseases, parasites, and symbionts. The relationship between phlebotomy sandflies and their host parasites and symbionts have been thoroughly reviewed in various books and publications, but these associations range from obligate heritable endosymbionts to human disease organisms for which phlebotomides act as intermediaries. So for the Leishmania, the association between phlebotomies and leishmaniasis, a group of human diseases, has been highlighted by recent outbreaks in areas such as Afghanistan, Iraq, Brazil, and India. These outbreaks have resulted in tens of thousands of new cases and thousands of deaths. There's 4 clinical categories: viscerocutaneous, mucocutaneous, and diffuse cutaneous. A standardized system of parasite identification based on iosome profiles have been developed with over 30 recognized Leishmania species. However, the identification of vectors for each Leishmania species is complex, with multiple phlebotomized species incriminated or suspected as vectors. Incrimination of phlebotomized species involves 4 steps: feeding on humans, sufficient densities for cyclic transitions, permissiveness to parasite development and multiplication, and transmission of the infectious parasite. So in some cases, a phlebotomized species may be guilty by association, indicating the vector. Distinguishing vectors from non-vectors is challenging in areas with high species diversity, requiring careful identification of sandflies and parasites. characterization of densities, and human feeding affinity and laboratory transmission studies. For bacteria and viruses, in addition to Leishmaniasis, phlebotomites also transmit other human disease agents, although these are generally localized or have low pathogenicity. One example is Bartonella, which causes Bartonelliasis in the Andean highline areas of Ecuador and Peru, with a high mortality rate in its virulent form. However, the sandfly species associated with this disease have not been conclusively incriminated as vectors. The phlebotomites also transmit variants of the vesicular stomatitis virus, an important pathogen of veterinary animals, and the Bungieviridae family, which contains 23 species isolated from the Old and New World phlebotomites. However, only 5 flavivirus species are known to cause human disease, and only 2 sandfly fevers have gained some notoriety. Now, for the symbiotic bacteria, so scientists use a DNA-based test to look for the type of bacteria called Wolbachia in 15 species of insects. They found Wolbachia in 4 species, 2 phlebotomists and 2 Lusmia. The types of Wolbachia found in each species were different, suggesting that they came from different sources. But only 1 out of the 5 field samples of Leishmania were infected with Wolbachia with a W, indicating that this bacteria is not highly infectious in these species. Now for the conclusions. So the study of diseases transmitted by sandflies will continue to be a rich area of research for years to come. New phlebotomite species are still being discovered, and the diversity of genetic variants among New World Leishmanias is only just beginning to be understood. This complexity makes it challenging to develop effective control measures, but new technologies are helping to advance our understanding of the interactions between vectors, parasites, and hosts. Molecular tools are being used to quantify variability and assess relationships among vectors and parasites, and PCR is allowing for rapid and accurate detection and identification of the parasite in hosts and vectors. The distribution of vector species is being mapped using GIS, which will provide initial assessment, a risk that can be scaled up as more data becomes available. As the distribution data becomes more dense, the accuracy of risk assessment and prediction will increase, allowing for more effective control measures to be implemented. Now, what has changed since the book was published? So discovery of new species, for example, the Sinuotonius africana, which was discovered in South Africa in 2019. 2 new species of phlebotomists were discovered in Brazil in 2018. And understanding of molecular interaction between sandflies and Leishmania parasites. So studies have shown that sandfly saliva contains immunosuppressive molecules that impair the host's immune response to Leishmania infection, and the development of genetic tools for species identification and control. So the next generation sequencing techniques have enabled rapid and accurate identification of sandfly species. Genome editing tools have been used to modify sandfly genes and study their own disease transmission, and genetic markers have developed to track sandfly populations and monitor the effectiveness of control measures. So these advances have significant implications for understanding and controlling leishmaniasis, a neglected tropical disease that affects millions worldwide. And that's it. Thank you.